Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to the Genetics Podcast. I'm really excited to be here today with Dr. Sarah Marzi, who is a senior lecturer at King's College London and group leader at the UK Dementia Research Institute. Sarah's lab is focused on understanding how genetic and environmental risk factors shape cell states and drive some of the diseases we'll be unfortunately very familiar with: Alzheimer's, Parkinson's, ALS, and neurodegeneration more broadly. Sarah's team and her collaborators work both on large-scale human data, but also diving into single-cell approaches and looking at multi-omics and all kinds of really relevant tissue types that we're going to jump into today. What makes this episode particularly cool for me is Joy, who produces the podcast, used to work with Sarah, and so my prep on this has been incredible because I have almost an inside look at some of the really exciting things you're working on. And we bumped into each other at ADPD, one of the big Alzheimer's and Parkinson's conferences a few weeks ago. So this is fun, very serendipitous, and it's a really exciting time actually in the field. So Sarah, thank you for taking the time to join me.
Thanks so much for having me, Patrick. And yeah, it's really exciting. I heard a lot about the podcast from Joy. I have watched some episodes that were really amazing. So it's an honor and a pleasure to be here today.
Great. I tell everybody that when I have great guests, it makes my job easy, and this is no exception. So I'm very excited for it. I mentioned your work connecting genetics and environmental risk. Just before we get into the details of some of your really interesting recent work, can you just share a couple of examples to help make this concrete? I'm thinking about pesticides and Parkinson's disease, or maybe the head trauma and ALS, FTD, that's maybe not so well established. Could you share a little bit about the big environmental risks you think of and what we know?
Absolutely. So also, I'd say the contribution of environmental factors varies a bit from disease to disease, and Parkinson's is one of the less heritable and less genetic diseases, with maybe only around 30% heritability. So meaning environmental factors are likely to play a bigger role. You mentioned different agricultural pesticides. I think there we have repeated and quite robust evidence that these are causally implicated in disease. Heavy metals, such as you might be exposed to in the mining industry, have pretty good evidence as well, as do industrial solvents and compounds that you could be exposed to in the heavy metal industry, degreasing agents. So that's Parkinson's. ALS, it's a little bit harder because it's a rarer disease, so it's harder to establish what is actually causally implicated in disease. And you mentioned head trauma, and there seems to be this association with some famous rugby players who've developed ALS. Although, again, that's not fully causally established because it's so rare. And there seems to be also a thing about elite athletes having a higher rate of ALS. But so we don't really know if is it the athleticism and the doing this extreme exercise that is predisposing you to ALS, or is there maybe an inherent factor that makes you both better at elite athleticism, but also gives you higher vulnerability to ALS? Other than that, I think there's things with comorbidities with other diseases. So, for example, in Alzheimer's, there's an observation that people with type 2 diabetes are at higher risk of developing Alzheimer's disease. So it can be a bit of a spectrum across diseases, lifestyle factors, and truly extrinsic environmental risk factors.
Yeah, really interesting. Do you have, what's the current state of our understanding of how much these environmental risks interact with genetics? It strikes me as something that's really hard to measure, just given the size of the data that you need and just the nature of the two different data types. What do we know, or what do we hypothesize if we're not sure yet?
Absolutely very hard to measure. So I think everybody agrees that there's at least an additive effect of both things, but whether that's multiplicative or that differentiates each other is much harder to determine, just as it's hard to determine whether there are gene-gene interactions and this concept of epistasis, where the variants don't just additively add up risk, but that their interaction in turn makes your risk that much higher.
Yeah, makes total sense. Okay, let's talk about some of your work. You've got a lot of interesting pieces of work across Alzheimer's and Parkinson's. For people who aren't familiar with your work yet, could you talk a little bit about one or two of the most exciting projects you're working on right now?
Absolutely. So I'll probably start with Alzheimer's and our work that we've been doing on microglia. So microglia are the resident immune cells of our brains. They're a type of macrophage, which generally means they eat up and digest different toxic things, debris that shouldn't be there in any tissue that they live in, but also in the brain. So for example, when neurons die and they leave sort of debris, leftovers from the dying neurons, the microglia would come in and clear that up. And microglia are very interesting in Alzheimer's disease because research in the past 10 years or so has shown that when you look at the heritability and the genetic risk associated with Alzheimer's, it very much is enriched in and co-localizes with regions of the genome that are regulatory elements that are selectively active in only microglia. So particularly enhancers that only exist in microglia and none of the other brain cell types. So that really suggests to us that whatever this genetic risk is doing that is making one person more likely to develop Alzheimer's than the other is changing their microglia and features and functions in their microglia. So really, we want to be studying microglia and what these functions are that make your risk higher or lower for Alzheimer's disease. And one of the strongest genetic risk factors for late onset Alzheimer's disease is actually the variants in the gene APOE, apolipoprotein E, which occurs in 3 different isoforms or 3 different variants. APOE2, which is actually protective, APOE3, which the majority of people in the world have, and APOE4, which gives you higher risk for Alzheimer's disease. And so we did a really neat study of microglia with these APOE variants. So basically genetically engineered microglia that only differed in the single APOE variant, APOE2, APOE3, APOE4. And to study them in a disease and whole organism context, we used a pretty amazing model that was developed by our collaborators, Bart de Strupe and Renzo Mancuso. So this is a xenotransplantation approach where you basically remove a mouse's microglia and inject human microglia derived from individual potent stem cells into that mouse brain. And you can do that at pretty high efficiency. So after that, about 80% of the microglia in that mouse brain will be human. And the pretty cool thing is that they behave quite normally. They look more morphologically, functionally. They express typical microglia genes, and they seem to interact with the other brain cells in the mouse brain. So you give it a pretty realistic environment. And by putting it into an Alzheimer's mouse model, you can explicitly expose it to the neuropathology, so these proteins that aggregate in Alzheimer's. And of the 2 proteins that aggregate, so you have amyloid, which forms plaques between the neurons in Alzheimer's, and you have tau, which forms fibrils within the neurons. And most of the mouse models of Alzheimer's disease only capture one aspect of these diseases. And so the one that we used is an amyloid model. So it only has these aggregating amyloid plaques. But it lets us study the microglia in this sort of protein aggregation challenge environment in which they should be helping to clear actually the early stages of aggregation of these proteins up. And that is something that we think that fails eventually in Alzheimer's disease, leading to this aggregation.
Why is it that the mouse models can only capture one of the 2 aspects of the disease? That's interesting.
So it's not that they can, but I think you have to, the genetic manipulations required to capture all the aspects of disease are so harsh that the question is, how realistic is that still, and how it's a little bit Frankensteinian. You're engineering all these different things on top of each other and making something that's probably not very viable for very long. And how representative is that then of human disease? So I don't think any of the mouse models are fully representative of human disease. And partly that is because mice don't normally naturally get Alzheimer's disease.
Right. That makes sense. Thank you.
Right. So that's the setup. We've now got the cool model that we can put the microglia in, and we've got the genetically engineered line with the APOE genotypes, and we can look at how the different APOE microglia respond to this challenge. And so we looked at gene regulation, gene expression, and epigenetics in these microglia, and actually found that there are quite some striking differences across the different APOE genotypes, including that APOE4 microglia are worse at moving around, so cellular motility and chemotaxis, which is the ability to sort of go to a site of injury or infection and do their job, seems to be impaired. They seem to upregulate pro-inflammatory cytokine release, so driving a stronger neuroinflammatory response. And so that seems to be a staged effect from the lowest pro-inflammatory cytokine release in the E2, medium in the E3, and highest in the E4. So, and finally, they also seem to be worse at phagocytosis. So this function where they eat up debris and different things. And we were actually able, for the phagocytosis, we're able to show that, in fact, functionally in vitro as well. And so we wondered, these are all the differences, but are there any sort of key drivers that we might be able to enhance or target and manipulate that are driving these bigger transcriptional responses leading to these functional defects? And so one thing that we did was transcription factor motif analysis. So basically where you look at the epigenetic changes that we observe, are they enriched in the binding motifs of very specific transcription factors? Because that might give you evidence that one particular transcription factor is having increased or decreased binding at these motifs and driving a whole bigger scale transcriptional response. And we were very excited to see that the motif for the vitamin D receptor was actually enhanced in the E2 microglia. So they seem to have increased binding of vitamin D receptor. What is vitamin D receptor? It's a nuclear transcription factor, so meaning a molecule basically that binds DNA and regulates transcription of lots of downstream genes. As the name suggests, it's activated by vitamin D. And vitamin D is really interesting in the context of Alzheimer's disease because there is some very robust evidence that vitamin D deficiency is a major risk factor for Alzheimer's disease. You have low vitamin D that both increases your risk for developing Alzheimer's disease, and even once you already have Alzheimer's disease, it worsens your progression, so it makes your disease course faster and steeper, if you will. And so that's really interesting because now we have this epidemiological evidence with a direct link to the disease, and this very molecular evidence linking a well-known genetic risk factor to the functioning, downstream functioning of vitamin D and vitamin D receptor signaling. And so we published this last year. We're super excited about it. We've got a whole set of in vitro and more xenotransplantation experiments set up to study that in more detail, understand the detailed functional consequences, and try and pharmacologically and genetically manipulate the function of vitamin D receptor to see, essentially, can we rescue or improve the harmful APOE4 microglia to make them more like the APOE2 microglia.
And so is there a way to think about the relationship between APOE status and vitamin D? Going back to this question of additive versus multiplicative risk, is there a naive take here that
so I think there is still a lot of functional mechanistic links still lacking. Yeah. We see this effect on the vitamin D receptor, but we don't know what's the actual molecular cascade of events. Yeah. One thing that is likely happening, so APOE is a lipid transport, cholesterol transport gene. And so one thing that we see in Alzheimer's microglia is this increase in lipid droplets, accumulation of lipids. And that in turn seems to correlate with this pro-inflammatory immune activation. So maybe the link goes that way. How that then links to vitamin D receptor, not fully known. I believe there is some evidence in the literature as well that actually APOE4 microglia do have vitamin D in them. And so maybe they just fail to activate the receptor, or the nuclear localization of the receptor isn't working properly. We also know it needs to heterodimerize with the retinoid X receptor. So it's not as simple as vitamin D activates the receptor and then everything downstream happens, but there's a lot of required steps. So it could be anywhere in that cascade that something does not work properly.
Yes.
The APOE4 microglia.
Maybe this is a good segue to talk about something you're working on, I think, right now and have been for a number of years, which is looking at postmortem brains, because the xenotransplantation is a really interesting model. But if you can come at this from another supporting angle by looking at human tissues, it's obvious to all of our listeners how hard it would be to do this in any system except a mouse model or a cell model or something like postmortem brain. So can you talk a little bit about what you're doing there and what you expect to learn?
Yeah, absolutely. So we teamed up with John Mill and Emma Dempster in Exeter to generate the biggest multi-epigenomic study of cell type specific epigenetic regulation in the Alzheimer's brain. So with a target of about 200 brain samples, 100 Alzheimer's disease, 100 pathology-free controls, but really going into cell type specific signatures, which is super crucial when you're looking at epigenetics, because these enhancer elements and epigenetic signatures tend to be highly cell type specific. So you can imagine when you just take a bulk brain tissue piece, that's a combination or an average of all the cell types that are contained in the bulk. And so really what you want to be understanding is what's going on in each of these cell types. And so we invested a lot of effort to purify different cell populations with flow cytometry. So you can use different nuclear markers. They'll often be transcription factors, actually, to purify different populations of nuclei, and then run epigenetic assays. So for this atlas in total, we'll have a TAC-seq for open chromatin, then DNA methylation data, and my lab in particular, and actually this was led by Joy, who's now running the podcast with you, performed cut and tag for the histone modification H3K27 acetyl. This is a specific marker of active enhancers and active promoters. And so we've now got data on up to 150 samples, I think, post-QC per cell type for oligodendrocytes, neurons, microglia, and also a triple negative population, which is sort of everything else, probably enriched for astrocytes and a bit of perivascular cells.
Wow.
And so that's an amazing resource because, as you say, in the mouse, yes, we can study the very specific effect of that one genotype, but it's kind of an artificial system. We only want to have one genetic background. That's a really important thing to consider. Things could look very different depending on the whole genome background because all those variants are going to have an overall effect. So here we have a lot more data. We have the real human disease and the real human sample. And we do actually see some really interesting things, for example, relating to lipid processing and lipid signatures in microglia that are upregulated and enhanced in all the Alzheimer's cases, independent of their APOE status, actually. And that relates back, I guess, to some of the signatures that we'd seen in the APOE. But not everything is super consistent. Always something that's also quite striking that we've seen come up is quite a consistent downregulation of type 1 interferon response. Now, this is normally a response to viral infections. And we're not entirely sure whether this is a consistent disease course response or whether another possibility is that we're collecting very late stage samples, actually with extremely high pathology burden, whether these microglia just eventually become exhausted and are not able to mount those immune responses anymore towards sort of the later stages of life and disease.
Yeah, wow. Two questions. The first is how much of this is like the microglia are the star of the show, or are there lots of things happening in a big cascade? What do we know about all like the role that all these cell types play?
Yeah, lots of things are happening for sure, and I think it's sort of a multi-cellular, multi-systems disease. Certainly in what you see happening in the brain, but then also in what's causing the functional deficits and the clinical symptoms of the disease, right? So eventually the neurons die, and that's probably what's causing the real disease symptoms and eventual death as well. But the genetics, again, has told us that likely the causal upstream culprit for that is variation in the way that your microglia function. Other things that you see quite early on is the loss of myelination. So oligodendrocytes are these quite fat, heavy cells that you have in your brain that insulate the axons, so the signaling tracts of the neurons. And that works very much like cable insulation in your or other electronics hardware. And so they make the signal conduction faster and more accurate. And when you lose them, you lose those advantages and you get signaling deficits and downstream probably loss of synapses, these electrical connections between neurochemical and electrical connections between the neurons. Another feature that happens potentially relatively early, and that's certainly not good, is that your blood-brain barrier, so your basically the blood vessels in your brain are protected by this layer built by different cells called the blood-brain barrier. And that's important because you don't want any infectious agents or dangerous things to get into your brain. And this sort of breaks down in Alzheimer's disease and becomes leaky. And so it's more permissible to things, both infectious, toxic things, but also potentially peripheral activated immune cells that could wreak havoc in your brain to come up and cause additional damage. So I think as the disease progresses, actually a lot of cells, probably most of the cells that we have in our brain, are in some way implicated and can potentially be harmful. But I guess ultimately for preventing or stopping the disease early, we do want to often anchor this on the genetics or on the very causal risk factor so that we can treat whatever is driving the whole cascade of events.
Yes. Yeah, what is your current thinking on therapeutic strategies? Because when you have this multi-cell type long-term cascade, there's lots of different times you can intervene. There's lots of different ways. What's your current thinking? There's a lot of people trying a lot of things. Where do you see the most promise?
It's so difficult. Where do I see the most promise? I mean, as you probably know, the only therapeutics that are out in the world now that are doing something molecularly and clinically are these anti-amyloid therapies. Often when given at these later point in diseases when you're already symptomatic, they do clear up the amyloid plaques, but they definitely don't stop the disease. They can slow it down for a little bit, but it's certainly not a cure-all. There is a big hypothesis in the field that is that the amyloid is the sort of onset event of Alzheimer's disease, and that everything else that is happening comes downstream of that. I mean, that remains to be fully proven. Maybe if you could intervene with these anti-amyloid therapies extremely early, and you can show that they might be preventative in such a way, then we have more evidence to that degree. But I think until we get there, we're going to have to work a lot more on treating symptomatic disease as well, especially because we can't really diagnose it pre-symptomatically yet. It's very difficult to find it. And actually, the disease will probably have been progressing in your brain 10 to 20 years before you get any cognitive symptoms. So that's really tricky. So I think for therapeutic strategies, I think we're probably looking at a cocktail of things that alter different potentially causal and upstream events, but also enhance function for longer. And that could be things targeting synapses and synaptic function that might both keep our cognition intact for longer, but might also enhance neuronal survival for longer.
Yeah, interesting. We've gone down the Alzheimer's branch of the tree. You have a ton of other work in Parkinson's. I'd love to hear a little bit about some of the big things you're working on there as well.
Great. Yeah, of course. So Parkinson's is, like I said in the beginning, a much less genetic disease. That's not to say that genetics isn't important. And in fact, the genetics of familial Parkinson's and very high penetrance variants has taught us a lot about, for example, the involvement of mitochondria and mitochondrial dysfunction in Parkinson's. But like I said, 70% of the variation in disease occurrence is not explainable by genetics. And so to me, that really shouts that we need to be looking at environmental factors a lot more. And the second really interesting and worrisome fact about Parkinson's is that the rates of Parkinson's are rising. The rates of all late-age neurodegenerative diseases are rising to a degree because we have higher life expectancy, and therefore diseases of older age are becoming more common. But with Parkinson's, it seems to be rising beyond what is explainable by just the increase in age. And so it's rising faster than some of these other late-age neurodegenerative diseases. And that, again, suggests that it's something in lifestyle or the environment that we're exposed to that might be driving this. And so things that are discussed quite heavily is sort of urban living and the factors that you're exposed to in conjunction with that. So air pollution, different types of particulate matter. They are discussed in all the neurodegenerative diseases. I don't think we have good causal evidence for any of them yet, but certainly they would be sort of common culprits, and for that reason alone, I think would be important to understand them better. In terms of our specific work, a lot of what we've done so far has actually focused more on the pesticides that we mentioned in the beginning, and that we've done that selectively because we wanted to work with something where there's credible causal evidence, because we want to really understand disease mechanisms. And my hypothesis is that with all this plethora of different risk factors that have been identified already and that we keep identifying, I don't think that each one of them has a very different, unique pathway, biological pathway that independently leads to disease. I think what's more likely is that downstream, many of them converge maybe on a limited number of biological pathways, and that by studying them, we can maybe understand those pathways. And maybe they also overlap with the more polygenic component of the genetic risk as well, or maybe they're all hammering down on a few of similar pathways, and you have some additive or interactive contribution of the genetics and the environment on those pathways together. And so we started out actually with a pesticide called Rotenone. This is a complex 1 inhibitor, so it works by as a primary mechanism of action by inhibiting the function of mitochondria, and in particular complex 1 in the mitochondria. But what we don't understand, so complex 1 is essential to the functioning of all cells in our bodies. And yet, when we're exposed to this as humans, or there are some animal models for this in the rat, for example, where you inject this at low doses, it's not all the cells that are dying. It's very selectively the dopaminergic neurons of the substantia nigra that die both in Parkinson's and in these Rotenone exposure modules. And so we want to understand why are they so vulnerable to this, and what are the functional changes, cellular changes that are happening in response to Rotenone, both in the dopaminergic neurons, but also the cells around them that are potentially interacting with them and contributing to this as well. And so for a lot of this work, we've teamed up with Tim Greenemeyer and Emily Rocha at the University of Pittsburgh. So Tim first developed this rat model maybe 20 years ago or so now, and has really been working on the role of toxicants in Parkinson's disease, and also asking the question whether they biologically converge on certain cellular functions and mechanisms. And so our first study that we published last year looked at basically bulk epigenetic profiles, both in the substantia nigra, which again is the area of the brain where you get the loss of dopaminergic neurons, but also in the motor cortex. And the motor cortex is interesting in Parkinson's, not because neurons die in it, but because it's downstream functionally affected by projections that first degree or second degree come from the dopaminergic neurons in the nigra. And so you would expect potentially functional deficits, lack of input in the motor cortex. And so what we saw in this study was, on the one hand, reassuringly, yes, we pick up that basically respiration and function of complex 1 and mitochondria is severely inhibited. And you can see this in the gene expression and in the epigenetic data, and that's consistent in both the motor cortex and the nigra. but maybe not so interesting because we already knew that's the main function of Rotenone. What's more interesting was when we tried to ask, so what's different between those two regions and what might explain why you get this neuronal loss in one of them. So in the nigra, you get a unique and quite heavy immune upregulation of both adaptive and innate immune pathways that likely come from the microglia. At this stage, this is just bulk tissue, so we weren't 100% sure in the paper, but we've been generating some single cell data in parallel, and it is indeed the microglia that are causing these signatures. And in particular, one of the immune responses that was so striking, upregulated by, 4 or 5-fold, and really only in the nigra, was the complement system and the C1q component, which is one of the upstream components of the complement cascade. And C1q is made up of these 3 chains, C1q, A, B, and C. 6 of each of them build C1q.
Wow.
And it's heavily upregulated just in the nigra. And when we look now at the unpublished single cell data, that is particularly in an activated type of microglia that shows sort of disease-associated interferon response signatures. And they're the only cell type that is really upregulating the C1q response. So we're not 100% sure how that in turn relates to the neurons. C1q in neurodegeneration more generally has been implicated in synaptic loss, for example. So it can be a signal that marks synapses for digestion by microglia again. And so maybe that is something that is happening, and maybe that is contributing to the additional vulnerability of those dopaminergic neurons of the nigra.
Yeah, interesting. Do you see, it's interesting to me that it seems like you don't see the level of pesticide exposure impact in Alzheimer's that is established in Parkinson's. Do you have any sense of why it would be so? Because it seems like the kind of thing that would wreak havoc in neurodegeneration more broadly. But do we see it in FTD or ALS, or is it seems to be a Parkinson's specific?
It could potentially. So people wonder about this all the time, and why is such a broad toxicant having such a specific effect on only one specific type of neuron? So one of the theories goes that it's about the morphology, the size, the energy intensity of those neurons. So dopaminergic neurons are heavily branched, very large, very energy intense neurons, and that in turn makes them particularly vulnerable to loss of mitochondrial function because they require more energy. However, I would argue some of the arguments that are being made here are the same arguments that are being made in ALS with the motor neurons of the spinal cord and the brainstem. They are also very large, maybe not as branched, but they have these very, very large, probably the longest axons of any neurons in our body. And that in turn requires a lot of energy to keep intact, to transport molecules, RNAs, proteins, and different things along. And so I think there is probably something to it, but we need to understand what are the differences between those, because otherwise, why aren't the same risk factors that are increasing our risk for Parkinson's at the same time making us develop ALS?
Yeah, interesting. I have a question that's been bugging me that maybe you can help with, which is you mentioned that we see the heritability as being higher in genetic heritability higher in Alzheimer's, lower in Parkinson's, but it seems to me that in clinical trials, it's almost flipped. There's a lot of LRRK2 and GBA trials ongoing or emerging. There's lots of idiopathic PD trials as well, obviously. But in AD, we see very little APOE4 targeting or other genetically targeted trials. Is this something that's going to change with time, or is the genetic signal there, but it's just more diffuse in Alzheimer's? How do you think about that?
Yeah, I think the last point is probably the pertinent point here. So what I sort of skipped over a bit when I talked about heritability is that I talked about the sporadic forms of disease, so the late onset forms that are not familial.
Yeah.
And actually, the familial forms are a bit more frequent in Parkinson's. So it's probably in the low single digits percentage in Alzheimer's, and it's caused only by 3 genes, APP, presenilin 1, and presenilin 2. And those are being investigated. But essentially, all 3 of those lead to more production of amyloid and thereby more aggregation of amyloid. Whereas in Parkinson's, you maybe have around 10 or just over 10% of familial cases, and they're caused by a bigger variety of different genes, both recessive and dominant, including variants in GBA and LARC2. And so there we understand more about their function, and therefore I think we have more clinical trials in that space. So there are more genes. There are about 15 or so with large effects in Parkinson's. Whereas the big heritability, the sort of 70% or so that you get in sporadic Alzheimer's disease beyond APOE is largely polygenic, so very, very difficult to target individual genes in that space. And I guess that's where my hope is, that the functional genomics, the epigenetics, can let us understand bigger transcriptional responses that might be modulatable through a limited number of transcription factors, for example.
Oh, yeah, interesting. So the polygenic, the polygenic background is going to hopefully collapse on some familiar pathways that we can simplify a little bit. Yes. Yeah. What do we know about that?
That's my hypothesis. What do we know about that? I don't think we're there yet at all yet. I think we've understood for a few individual variants what cell type they act in and what gene they might target. I don't think we are starting to find the transcription factors that are driving specific cell states of microglia, and that's super important and interesting. But I don't think we fully deeply understand, and I don't know if there's an answer to whether there are good protective cell states and bad cell states, or whether that's all adaptive and context dependent. So a cell state could be useful in the early stages, but then become maladaptive later on. So it's very tricky and potentially requires quite a bit of fine tuning.
Yeah, interesting. When you go, naive question, but when you're looking at something like the postmortem brain tissue that we were talking about before, what constitutes a cell state? Like it seems like you can coarse grain it, fine grain. There's lots of ways to think about how many states do you want to have, and how detailed do you go? How do you think about that?
Debate. Yeah. Very open debate in the field. I think cell types are pretty clear. Cell states or cell subtypes, much more open debate. I think the biggest advances and the biggest impact there has been made from the field of single cell RNA sequencing. So just sequencing lots and lots of cells and looking at their transcriptional profiles, and often agnostically clustering that. And but again, you need to make some decisions about how many clusters do I allow, and you can subcluster and subcluster ad infinitum. Basically, you can keep doing finer grained things. And actually, when you do see that cell states aren't so much a discrete thing, but they're more of a continuum. Right. And there potentially are also trajectories going from homeostatic in microglia, for example, to more activated states. And there might be sort of 2 or 3 maybe different arms that they can go along.
Yeah, really interesting. We're running out of time here. This has been an amazing conversation. I have 2 more questions. First one is just about any other interesting work that you're working on right now that you want to make sure you share or cover. You have quite a few different things going on, so it must be hard to keep track of it all. But yeah, and anything else you want to cover before my last question is more of a fun one.
We do. So I'm going to stick with AD and PD for now, because I don't think I want to introduce a whole nother disease. But yeah, some of the more recent work also in the postmortem Alzheimer's study has really highlighted that actually the largest number of epigenetic and potentially also transcriptional changes aren't happening in the microglia, but in the oligodendrocytes. And this is something that had initially surprised us, but that seems to be quite consistent across now 4 different data sets or so that we've looked at, and it's always the oligodendrocytes. And so we were just awarded some funding with collaborators, including Alexi Knott at Imperial and Dervaselli at UCL, to really look at that more deeply. So again, I don't think there is any genetic evidence for the oligodendrocytes. We do know that there's functional deficits and loss of their myelination function in Alzheimer's disease, and that's probably a bad thing. But yeah, we were surprised and excited, curious to see that they have these really massive changes. So they're remodeling transcriptionally, epigenetically, very heavily in the disease. And so we want to understand whether that has an impact on disease outcomes. And again, whether long-term that is something that you can potentially target to improve functional outcomes.
Yeah, super interesting. And we didn't even talk about, you're also doing work on astrocytes, right? There's lots of glial cells doing the neurons get all the attention, but the glial cells are doing all sorts of important stuff.
Yeah. Super interesting.
Okay, last one is just a fun one. My understanding is that you're a big hiker, adventurer. I want to hear about some of the most memorable treks that you've done.
That's a great question. Yes, I'm such a cliche German. I love hiking.
That's true. Yeah, it's great. It's a great cliche.
About 2 years ago, and I know this because I get the Google photo reminders these days, I went on an amazing 2-week trek in the Himalayas in Nepal, and it's called the Annapurna Circuit Trek. So it's not climbing Mount Annapurna, but it's sort of doing a horseshoe-shaped 3/4 turn around it. Cool. And as you do that, you walk over the highest mountain pass in the world, the Thorongla Pass. So that I think was one of the most incredible experiences ever. It's so beautiful. It's so humbling, but not in a depressing way. When you're in front of 8,000er peaks, you definitely feel small, but in a sort of beautiful and liberating way, I think. And so would recommend that to anyone.
Did you grow up doing lots of hiking, trekking, or how did you get into it?
Well, I mean, yes, a little bit. I was in the Scouts, and camping and some outdoor activities. I was also at some point, like many of us, a moody teenager who thought hiking was only for boring old people. So I think when my parents wanted to take me on hikes, it definitely wasn't there. But I'm in my 30s now.
And you're finding your hiking era.
Yeah, no, really, really love it now.
Amazing. Well, Sarah, thank you for this really great interview. I really appreciate all the time and congratulations on all the great work that you're doing.
Thanks so much for having me. It's been a great chat.
My pleasure. Thanks, everybody, as always, for listening, and we'll see you next time. Thanks, as always, for tuning into the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sonogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sonogenetics, on LinkedIn, or reach out on Instagram at The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show. Jane Reddy-Webster keeps us organized, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.